Hey guys, welcome back to my channel, the series of learning how to R from nothing. Today I'm going to show you one of the most critical initial step in analyzing any kind of single-cell RNA sequencing data, whether you have multiple sample, donors, conditions, using one of the most popular R packages out there, if not the package to analyze single-cell RNA sequencing, that's the Seurat package. So let's get started. And you might notice that there are several thresholds during the tutorial, or not even just tutorial. Publications sometimes just tell you, for this particular QC metrics, I choose this threshold, but they don't really explain you why or how they come up with the threshold. So that, as a beginner, very frustrating. And of course, it's actually is in most cases, you shouldn't use whatever the tutorial or someone else's threshold on your own dataset, because guess what? QC, your dataset, is data specific, cell type specific, and so you really have to go through your data and decide whether this threshold makes sense. So how we do it? Today I'm going to show you exactly how you should, and different tools, different visualizations that you can use to assist you with deciding whether the threshold that you're going to choose is appropriate for your data. So the Seurat pipeline, there's many other steps that come before you get your data that is then the input into the Seurat Seurat package. That's a CellRanger, for example. And during the CellRanger, before you get your Cal matrix, they did already have some sort of QC, which we will not touch on today workshop. But it's worth taking time to go through the CellRanger pipeline, which is available on the 10x genomics website. So once you have your Cal matrix, which is basically a data table containing rows and columns, the columns being the cells, each individual cell will be one column, and each row will be a gene or what they call a feature. So once you have your Cal matrix, in this case, we'll have UMI. Again, the theory behind UMI approach sequencing, I'm not going to explain it, but it's abundant on the internet. So you can Google UMI and see why do we need UMI technology. Now, so the data that we actually will be using today is coming from PBMC, available from the 10x website. That will be 2,700 single cells from the peripheral blood mononuclear cells of humans. And so again, once you have this Cal matrix, you'll be able to input that into Seurat. And as I mentioned, the first step of QC in your data is to remove low quality genes and cells. Then from there, once you did perform the QC, then you are ready to do some normalization approach. There are various ways you can normalize your data, scaling, centering, then you apply some statistical analysis to identify cell-to-cell highly variable genes that you will then be able to reduce the number into most critical gene set using dimensionality reduction like PCA. Then you use those PCs of the gene set of interest, then you'll be able to cluster the cell using their transcriptome. And then finally, most people do downstream of clustering is define the gene markers that characterize their cluster cell of interest, and then you do the cluster annotation based on the DAGs, which is the differentially expressed genes, or from some automated tools that we might be able to cover in this particular workshop. So today, because I only budget this to be an hour or so, so we're not going to go through every single thing that you're going to see on this slide. But I just want to mention that, the QC process is an iterative process. Sometimes you have to, once you finish your dimensionality reduction, when you finish your clustering, you might have to go back to your initial QC step and perform then pick another threshold, see whether the threshold that you pick initially actually makes sense, or whether there's some step in the analysis pipeline that you need to change because of the potential confounding factor that you might have missed during your QC, but now you are able to identify them after you have your cluster. So just don't think of the QC process. It's a very linear process. It's a very iterative cycle that you might have to go through several times to get and decide what is best for your data. So let's get started with the QC. There will be two levels at which you do the QC before you get the cluster and after you get the cluster. So I'll show you both today. All right. So in order to follow through with me on the codings, I deposit all the codes and the little online notebooks on my GitHub page, which you can access by going to this particular URL here. So the way to download anything from this kind of workshop from GitHub page is that you come back to here, then there's a little button here called codes. You click on that and just download the zip. And this, so let's just demonstrate you. So download that zip on wherever you want to store it. Then once it's finished, okay, hurry. Okay, unknown time left. Oh, okay, great. That was fast actually. So then you just unzip the file, and in this folder you find everything that you need for your for today workshop. And in this particular folder, you find that I have a PowerPoint presentation, which I just showed you the first slide. And you also have the R markdown file of all the codes that we will be using today. And just open that with RStudio. Okay. There will be a folder here called Filter Gene BC Matrix, and then there's a subfolder called HG19. So you need to get the path directing there. So in order to get that, right click if you're on a Mac, get info, and copy and paste this particular directory as a path name. Then you go back to your code. I'll show you as well. So you go back to your code in RStudio. Okay, and you basically replace this directory, which is here. Okay, and just paste and make sure that you need to direct it to this particular folder. Okay, HG19, and don't forget, because I already have all the packages installed, so that's why I have this section of install packages comment out. In your case, if you want to rerun the code, just uncomment this whole green block of text and then run this whole thing, okay, to install and load the package. All right, so load this. Okay, so this will take the data that you just download and and then it's input into your R environment, which now become PBMC.data. Okay, and in order to, so you can run this code to see the data set. And here is, again, I told you that the input data is a count matrix with rows and columns. It's always the convention is that there will be the rows. Okay, so you have 32,738 rows, which means you have 32,000 or so genes and 2,700 cells. All right, and you can even just kind of see what the data look like by doing that. Let me put this one down here so it doesn't erase our input. Okay, so this is how it looks. You see these dots, most of them are dots because RNA single-cell RNA sequencing is very, very sparse data. But for some, you see number 1 and sometimes number 2, the counts. These are your count of the RNA molecules. So for example, for this particular gene here, and for these particular cells, there's one more RNA molecule that was counted for that gene for this cell. Okay, so one of the ways that I would like to start with my RNA single-cell RNA sequencing QC is usually when you start your experiment, you have some set of genes of interest because of your research question that you ask, and you if you just want to see whether the genes are present in your data set to start with before doing any QC, what you can do is just make it into a vector of genes. So in here I call them interesting genes, and they look like this if you run that code. So I just pick some random genes here as an example, and then you can run this particular code to see whether they exist in your data set. So in my case, all of these genes are present in my data set, okay? But we don't know how much of it is expressed, but we do know that they at least present in our data. The next step in our QC process is to filter out the low quality genes. Okay, so what do I mean by low quality genes? In Seurat package, there is a parameter called min.cells. It's basically say that this is the threshold that say, so let's say if you do min.cell equal 3, that means that we'll take all the genes that at least express in at least 3 cells. All right. So the reason being is because if you have a gene that is only detected in a very low amount of cells, then maybe that could be due to it being a low quality genes of your sample, and you won't be able to say anything meaningful about that gene anyway. All right, so one of the way to visualize your data set in order to pick the right threshold for min.cell parameter is using this histogram, which I have customized the code here for you. So you can just basically replace your data set with your own data with this PBMC data, and then you run this code here, which I already ran, but we can rerun that. So you see here, this telling you that a lot of genes, you can see the bulk of your data is actually below 0.5. Now this is on a log scale, so that means you need to take the inverse log. So that's inverse log 10 of 0.5 is around 3. That's 3 cells. So what this graph telling you is that most of your genes actually express in less than 3 cells. And again, this is actually very typical for single cell RNA sequencing due to the technology at the moment. So don't freak out that you're gonna set a threshold here and then you're gonna eliminate most of your data, which is actually what typical in single cell RNA sequencing. All right, so so then you ask me, well, why not like 4 cell? Why not 5 cell? Why not 10 cell? Well, the answer is not that straightforward, and the answer is it depends on your data set. You could have picked 10 cell, you could have picked 5 cell, but the key thing to remember here is whatever the threshold that you pick, you need to give the reviewers or people the rationale as to why you pick it. So what you can do, and what I show you, one of the way that you can do is actually check what you have filtered out. So what are the cells, what are the genes containing cell that you filter out by setting this threshold, and whether this cell actually have all the QC matrix, which we will go through in the tutorial, that make them really low quality cells. So that's how you QC them. But these genes, because they only express in a very low number of cell, whether it's 3 cell like we pick here or 5 cell, that is a very low number of cell for the expression of a transcript. So they will definitely be something that you don't want to include in your data. One of the way that I like to do, if you want to be more statistically sound, what you can do is compute the quantile values here by this particular code. And what you can do, if say your experiments contain multiple donors from multiple samples and you want to keep the QC metric consistent, and if again this is applied when your experiment only involve the same cell type, okay? So like for example, if you sequence the same T cell, they all T cell, they all B cell, or they all blood, they all PBMC, they all, there's some central similarity there of the cell type composition between sample to sample, then you can likely will be able to use the same quantile values for a threshold, instead of saying I'm gonna cut out all the genes that express in less than 3 cells. Well, you can now say we will cut out any gene that don't pass the threshold of 60th percentile of the values of the data. So it just a bit more consistent. And now once you decide this quantile value as a threshold for your first sample, you might likely be able to use the same threshold of a statistical metrics for the rest of your sample in your dataset. So that's just a better way to do it than just with a constant number. All right, so this is actually the code that you will need to use to set the min.cells, which is the gene threshold for your data in Seurat. So when you run that, it will filter these low expressed genes. And now what you ended up with is, say here now PBMC is a Seurat class object. It contains 13,000 features across 2,700 cells. Now as I mentioned, we started out actually here with 32,000 genes, and now after this QC you have 13,000. That's almost 1/3, is only 1/3 remain, and that's the typical in single cell RNA sequencing. All right, and now what you can also do is run this code to see whether the gene of your interest is still remain in your dataset after this first QC. Well, in my case, the last gene in this vector of gene of interest, which is called S1PR2, is no longer present in my data. Okay. So it's just some useful information to know. And another way that you can also examine what you got from after the QC for this step is you can ask how many cells actually that were that this a particular gene, in my case I've just picked FOXP3, that will actually have any detection at all of their RNA transcript molecules. So you can run this code, just replace with your gene of interest. And in my case, it say that 11 cells have 1 RNA molecules detected for this particular gene FOXP3. So if you're interested, that's how you do that. And you can, again, this is probably the most important step for this QC, is you want to actually get the cell that was actually taken out. Okay, so in order to do that, you can run this code where you don't set anything for the parameter min.cells. So basically you take all the genes, okay? So you run that, and then now you can again ask whether the gene of interest should be there, and they should because you haven't filtered out anything. And now you can run this code basically saying which cell in my dataset have the expression of FOXP3 greater than 0, and this will give you the cell barcode that you can then use to pull out these particular 11 cells to further examine them in detail, whether to see whether it's sensible to actually take out this 11 cell, given that they, for the FOXP3 gene at least, they only have very low expression, only 11 out of 32,000 or so cell, right? All right, so that's very good. The next step after filtering out low quality, lowly expressed genes is to filter out low quality cells. So how are we going to do that? Well, there's a tons of metrics that people use to filter out the low quality cells. One of them is percentage of reads that map onto the mitochondrial genes. The reason being is because cells that are dying or in the process of dying or death, they tend to release more mitochondrial content. So therefore, you expect that these dying cell or dead cell, they will have higher reads that map to the mitochondrial gene transcript. Okay. So the way to compute percentage of these for mitochondrial genes for your dataset in Seurat is by running this particular code. So just run that code for your own data. Then we can compute the percentile values to keep everything consistent if you want to use the same threshold for your order sample. And you can, another statistical matrix that is quite useful also to take is to find the mean, the median. The median is the same as the 50th percentile, okay, by the way. As always, one of the critical things about QCing is you always want to visualize your data. And then you see whether this threshold makes sense or not. Okay, so the way to visualize quantitative metrics like percentage of mitochondrial gene expression is through the violin plots like this, for example. Here you can see very clearly that most of your data actually have very low content of mitochondrial genes. Most of them have below 5%, but there's some cells that have very, very high content of mitochondrial, about around 20 as well. One cell here actually have more than 20% of mitochondrial gene expression. So the decision again now, whether we're gonna pick 5 or we pick 10, 15 is really up to you. But the key thing to decide whether 5 makes sense or 10 is better or 15 is again pick a threshold, and based on the visualization of your data distribution, and then pull out the cell that you actually filter and see whether these cells, whether they express all the genes that may be of your interest, or whether they are actually low quality that you don't really want to include in your data set. Okay, another way that you can also visualize the percentage of mitochondrial is by using the ggplot package like this. Okay, they give you the same information, and you can see again is that most of your cells here, if you pick 6, I think it should be okay as well, 5, but the key thing again is visualize your data to see the threshold that you decide based on the visualization makes sense. Then you can compute these various, statistical things used to make it more consistent and statistically sound if you're gonna use the same threshold for your order sample. Okay, so let's run this again. All right. So again, this is an exercise for you that you can do. Given all the tools that I already cover, compute the quantile values, compute the mean, median, and max, and plot the data with the VlnPlot or ggplot. And this I will go through another function in the Seurat package that can visualize 2 QC matrix at the same time. And this is actually very critical. One, besides visualization of your QC matrix, it's important that you don't rely on one QC matrix alone to decide the threshold. You need to visualize the different QC in combination to see whether the threshold that you set for one matrix makes sense for the other QC matrix. If you visualize them together, that will be very clear. So let's go to an example of that. So you can do this particular exercise on your own time, and the solution is basically down here. So again, another matrix that is very useful in QC your single-cell RNA sequencing data is called nFeatures in Seurat, and this is basically the number of cells per genes that you detected. So again, you can compute the summary of the statistic of your data distribution, and as always, you need to visualize your data. So this is the code that one way that you can do that, and you can see here from this data set, you can see that most of the bulk of your data is below 2500, okay, number of genes, and this is a very low one. Here is the lower limits of 200 values here. So then you can say, well, I'm going to take all of the cells that have at least 2500 genes and have at least 200 genes as a lower cutoff. All right, and again, the decision is up to you, but you need to visualize your data to see if it makes sense. All right, and so one of the ways to check that this actually makes sense, as I mentioned, is to visualize this QC metrics of the nFeature together with something else like percent.mt, for example. And you can do that with the FeatureScatter function in Seurat, and this is one of the way that you can visualize them. So the first plot, I'm going to visualize percentage of mitochondrial gene reads against the number of genes or nFeatures. And you can also, another metrics that you can use is called nCount that I didn't have time to mention. It's basically the number of UMI counts per cells, okay, that per gene that you can visualize. So you can visualize this combination, and see whether the matrix that you set for your nFeature and percent.mt makes sense. So for example, here, as you can see in this two plots, the very high percentage, the cells that have very high percentage of mitochondrial gene expression also tend to have very low number of nFeatures. So that's why it makes sense to filter them out for your percent.mt greater than 5. You could have set the threshold at 15 if you want, but then you will include these cells that tend to have greater than the bulk of your data. And again, mitochondrial genes can be an indication of active process of, and some paper even show that mitochondrial gene expression or cell cycle gene can be useful to determine cell type or cell state. Again, the decision is yours to set where you want it, but it needs to at least visually make sense for your particular data. And you need to ensure that the things that you take out are not actually contain some biological insight and they actually low quality. And this is very useful metrics here. For example, we remember that we set the threshold for nFeature to be 2,500. So does that make sense? Well, if you visualize that together with ncount, you see that these two QC metrics are very correlated. And the higher the nFeature, also that cell also have very high ncount. So this makes sense to set the threshold here, or you could have set the threshold to 2,200, and that would have been fine. And also, all right. So regardless of whatever you do, you need to have a justification for why you do that. And the best way is to visualize your data distribution, visualize it against different QC matrix to justify your threshold. All right, so this is a very critical step here as well. So after you see, oh yeah, this QC matrix threshold makes sense, what you can do is see how many cells that you would actually take out if you apply all of these QC metrics. In our case, we're gonna take all the cells that have percentage of mitochondrial gene expression greater than 5%, anything that below 2,000 gene, and anything that above 2,500 genes. And with that, we'll take out 60 cells. So I mean, in the grand scheme of things, just a logic check, reality check. Of the 2,700 cells, you take out 62 cells. That's reasonable to me. All right, so this is the actual function that you will subset out your cell using the QC matrix thresholds that you just decide. So you run that, then you get PBMC. Now you can run your PBMC and see that the cell that you get at this stage. So again, you see that it's go down from 2,700 to 2,638 cells in your data. As always, please save your data, otherwise you have to rerun everything up to now. All right, I also provide you with the code. So this, just to make the workshop a little bit more interesting, I also randomly assign each cell to a group. Like I call it group 1, group 2, or group 3. You see why I do that later on, but just run this code as well if you want to follow along. It's basically we'll create a column called groups in the metadata of your Seurat objects that assign randomly group 1, group 3, or group 2 for your cells. All right, so this is the code where you can use to summarize what you have done after your QC, just to see how many cells in group 1, and what's the mean library size, what's the mean of the number of gene detected, that's the nFeature. Okay, so you can use this code, it's quite nice. And again, you can always check whether the gene of your interest that we set initially is still present. Again, they're all present except for the last gene here. Okay, and another way that you can also check your QC threshold is to say how many cells did I take out, how many cells actually expressed in my object after QC for a particular gene. So for example, you can ask which cell in my object after the QC that express BCL6. So you can do that code and it says 76. So in your data after the QC, you have 76 cells that have BCL6 transcript greater than 0. All right, of course, there are other QC metrics that we won't be able to go through all of them, but I mentioned them here, and some information and that you can further investigate your data, including sequencing depth, something called novelty score, or some people call it complexity score. Instead of mitochondrial and or mitochondrial gene expression, you can also do the housekeeping gene, ribosomal genes, that kind of thing. Another very popular topic in single-cell RNA sequencing is the doublets. Now, I decided not to use this metrics because the current packages that use to predict doublet based only on the transcriptome, that is the RNA-seq, is not that robust, especially if you have data that is very homogeneous. For example, if you sequence only B cell, and because the transcriptome between this cell tend to be quite similar in that regard because they're the same cell type, for example, the package, the way that it's work will because it only look to the transcriptome. This transcriptome might not be different enough for the model to separate out these potential doublets, or whether it's a real it's a real and not artifact in your data sets based on the transcriptome. But you can always check it out. There are two popular packages that I recommend that you can start with, called doublet finders, and another one is called scRNAseq. scRNAseq is a Python package, okay, and doublet finder is the R package. So you can work with either environment that you like and see. What I would suggest you do is use the manual QC, and then also run these packages to see whether the cell that you filter out based on your manual QC will actually predict as the doublets. Then you have an order rationale as to why you filter them out. All right. And another potential confounding factor is the number of cells in each of your condition, for example. Like, for example, here, between group 1, group 2, group 3, let's say you have 3 donors or 3 groups. And in group 1, you have 10,000 cells. In group 2, you have 500 cells. In group 3, you have 100 cells. So obviously, the amount of RNA that can be detected will be drastically different between these 3 samples. And now it's a decision for you to make whether you want to adjust for this difference in sampling. It could be sampling error that will lead you to some insight of the result of your data that might just be because you were not able to cover all the cells for a particular group. Okay, so here is another one way to visualize the amount of cell between different conditions in your data set that you can freely use as an example. And so, of course, more recently, people have come up with a modeling approach to help you decide the QC matrix. And one of the packages is called miQC. I put the link here as well, that you can use. What I would suggest you do is still visualize your data, go through the process that I presented today, and then do this package to see whether the threshold that you pick based on manual QC of your data, does it how much of this match with what the automated QC threshold that the package, the modeling gives you. And then you can compare and contrast. I wouldn't just use whatever the threshold that the automated QC tool gives you, but I would do it as a secondary validation. All right, so as I mentioned earlier from the beginning, there's 2 ways, there's 2 levels at which you can do your QC. You can do it before normalization. You can do during normalization. I like 3 level. You can do before normalization, during normalization step, and after you have the cluster. So during normalization in Seurat package, you're free to read this theory that I put here. There's 2 normalization methods that you can use. I suggest that you heavily look into this particular one because it's been shown to improve your data, especially for lowly expressed genes. It's called SCT transform here. I put some information that will definitely be useful to you to read. In this particular normalization, you have something called VARS to regress, and this is when you adjust for a particular variable like percent.mt or whatever in your QC metrics that you compute. Okay, so and then at the level, once you have the cluster, you can also adjust for during finding the differentially expressed genes, you can also adjust for that for a particular variable during that step as well. In the find marker function, which is the function in Seurat to find the DEGs, and there's a parameter called latent.vars, so you can set that into a parameter like, for example, your percent.mt as an example to adjust for that variable while you finding the differentially expressed genes. All right, so now that once you perform all of your QC, you're ready to do normalization, dimensionality reduction, and clustering. So once you have your cluster, I also put the way that you define your cluster in Seurat is through this process, which I won't go in depth. In theory, you can read it up on their website. But essentially what you need to do is you need to normalize it, run dimensionality reduction, run UMAP visualization, compute a graph, because this is a graph-based clustering. Okay, and in here, this is the step that I want to focus on. Once you find the graph for the clustering algorithm to detect the cluster, there's something called the resolution. This is the clustering resolution in the function called find cluster in Seurat package. You can pick 0, 1, 2, blah 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 blah. So how do you pick the right resolution cluster? Again, the higher the resolution, the more cluster that you're gonna get. The lower resolution, the less cluster number, the total number of cluster you obtain. In my opinion, it's always good to do over clustering first to start out, because you can always merge your cluster later on. Okay, versus if you're going to do under clustering, then you might lose the heterogeneity of your data set. Okay, so the way that I suggest you do, one way is you just make a for loop like I do it here of this function for different clustering resolution. Okay, so just compute that values. So let's do that. Hopefully it's not that long. Okay, so it shouldn't take too long to compute. So basically what you're saying here in this for loop is find the cluster for resolution from starting from 0 to 1.5, and each step plus 0.5. So it's going to compute the cluster with resolution 0, 0.5, 1, and 1.5. And what you ended up with is another column in the metadata of your Seurat objects for the resolution of 0 here, 0.5, 1, and 1.5 somewhere. Yep, 1.5 here. Okay, and this is the cluster ID, cluster 6, cluster 0, cluster 3. Now your task is to decide which cluster resolution makes sense for your data set. So the way that you can do this, one of the ways that you can do this is you can use a package called the clustree, I think is what's called here, clustree package. And this is one of the so if you run this, okay, let's see the graph that is produced. Oh, it's lose that, so let's do this one. So I'll explain to you what this graph telling you in a second. So if you run this code, by the way, you're free to use this code in your own data, just replace with your own objects. So what you see here is essentially is graph each of the row, each of this level here. As you go from the top to the bottom, there's increasing cluster resolution. So you can see that going from cluster resolution 0 to 1, you have these many cluster from to 0.5. From 0.5 to 1, you have these clusters split in 2, this cluster split in 2. But these one here are very, very stable cluster. They stay as one distinct cluster. So that's one way to visualize and help you pick the resolution. Another way that you should also take a look using this same package is if you have a number of genes of interest for your research question that you want to ask, you can run this line here, this code here. to visualize the amount of expression for that particular gene or set of genes. So let's run that. So what you get is this. So again, different level is increasing from resolution. And for example, here you can say that if I go from cluster resolution 0.5 to 1, this you can see clearly that, oops, sorry, hold on. All right, so again, as you go down the level here, so you can very clearly see that this one particular cluster very uniquely expressed the gene. I just pick a random gene called PBMC, and then you can decide that because going from cluster resolution of 0.5 to 1, this particular cluster still have very highly expressed gene and they don't really split into anything. So if this particular gene is of your interest, of your research question, then you can pick cluster resolution at 0.5 and that would be okay to detect it, because at this cluster, this cluster, this gene is still highly uniquely expressed in this stable cluster. All right, so that's good. So that's one way that you can pick the cluster resolution. And that's what actually I do in my own research. Now, and then you can, this code is to help you summarize how many cells in this is the code that you need to run to pick the cluster resolution. Again, in this case, we're going to pick cluster resolution 0.5. So you set the cluster ID on to this cluster resolution. Then you can ask how many cells do I get from each of the cluster, or you can just simply run this code instead and say active ident. Okay, once you set the identity of your object, then you see that the cluster that we just showed earlier, oops, now it's gone. Let's run add is at the resolution 0.5 is cluster 8. So that cluster actually have 12 cells. All right, cool. So once you have performed the clustering, your job as a QC, as QC is not finished. It's actually just started. Once you have the cluster, it's important to identify whether there's any potential confounding variable that you can detect by visualizing your cluster. Okay. So again, dimplot is a function that you can visualize your cluster that you obtain. So you can see they're quite well separated. And cluster A, although it's very small cluster, is a very unique cluster based on the transcriptome on this visualization. So you can visualize the different metadata variable that you have in your data. In order to access what meta variable you have compute, you can just run PBMC at meta.data, and then you can just get the column of them. So call names, because it's basically a data frame. So then let's make it up here. So run that code. So you have original identity, you have the end count, you have the end feature, you computed percent mitochondrial genes, and you have the group assignment. All right, that's weird because you should because this is the object before you have before you normalize. So you need to put the object with the cluster identity. Okay. So then see, you have all of your cluster assignment at different clustering resolution. So let's say you want to see, you want to visualize the cluster by groups ID. So then you can see that all of the cells are quite distributed. So there's not really there's not at all bias between the groups for your cluster. So then you don't have to worry about your groups. Based on this variable alone, you can just merge all of your data instead of having to integrate by group, for example. So that's one way to help you identify any confounding variable that might play a role in contributing to your cluster assignment. Okay, another function that you can use to visualize after you have the cluster of the quantitative metrics like percent.mt, because it's not a character, it's not categorical like the group assignment. Okay, it's numerical. So then you can do FeatureScatter. You can see that the percent.mt is well distributed across all of your cluster. There's some cells that have very high percent.mt, but not crazily in that particular cluster set there. So then you don't need to worry about percent.mt. Okay. Now, after you have your cluster, after your QC your cluster, now you can, I'm going to quickly go over how you can decide which cluster belong to which cell type, called cluster annotation. One of the way that you can do this, I thought this is very interesting way, is in Seurat package, you have this function called CellSelector. So if you run this, okay, then this would pop up a shiny app where you can visually, graphically decide which cluster you want to focus on. Let's say we want to focus on this cluster here, and you just make a box around it and then done. So then what it does is that it create an object called cluster4 cells, and this object will contain this particular cell that you visually select on the graph, on the UMAP graph. Okay, so then what you can do is perform differential gene expression analysis to see what is the gene marker that define this cluster 4 among all the cell in your data set by running this code here. So you run that, we run, and you see that the top DEG for this cluster 4, actually, or this cell that we visually select, is actually cluster 4, is MS4A1 and CD79A. These actually mark a B cell. Okay, so you can then visualize these particular genes on using the VlnPlot. We already know how to use the VlnPlot, but here again is the function. You can see that cluster 4, which is belong to the cell that we selected, actually have very high expression and quite uniquely of CD20. Basically, it's the gene that encodes for CD20 here. And then you can visualize 2 genes at the same time, and also see what cell actually have both of these genes is expressed by using this function called FeatureScatter. Okay, and you can see that again, this cell of interest here down here, which is cluster 4, are the cell that mostly express the genes that belong to B cell. So you have very strong evidence to suggest that cluster 4 in our case is a B cell. All right, so this is the last exercise for you. You can use what we have learned today using this very tool, the VlnPlot, dimplot, find marker to find your DEGs. This is to find your DEGs, and length and which cell function to pull out cells per according to a condition. Okay, and you can say, you can ask like, how many CD8 T cell in your data set compared to how many CD4 T cell in your data set. Well, I hope this is helpful to you. Let's go back to the PowerPoint now. So we did some coding. So this is the best QC practice in summary. In summary, again, QC is a data-specific and iterative process. Depending on the cell type that you expect in your data set, the amount of heterogeneity in your data set, that you need to decide which threshold, whether it makes sense or not. Always visualize your data differently and see which threshold makes sense when you combine your different QC metrics. Automated tools can be useful, but it should only serve as secondary, as a guide to cross-check your manual curation of your QC threshold. As mentioned, you need to use combination of QC matrix to filter your own data. And for cluster annotation, you should use biological knowledge. And also there's automated cluster prediction tools out there that we won't be able to go through this in this workshop. But okay, and I also put some useful resource about the Seurat package, the Seurat object as a class, and there's a paper, the very nice paper, detailed the various steps and consideration about filtering out low quality cells that you should definitely take a look. There's all the workshop that you can check out as well. In here is 2 examples. All right, well, that's it for me. I hope it was helpful to you, and I hope that you now feel more comfortable about how to go about setting your QC threshold for various QC metrics in your own data single-cell RNA sequencing using Seurat R package. See you next time.